Sean. What's up?
Hey, Alex, how are you?
I'm doing well. How are you?
I'm in trouble.
What's
I
wrong?
was stuck in the traffic. I couldn't make it. I missed my lab. So
No.
please tell me what happened in the lab.
Well,
What you did in the lab.
well, today in lab we did PCR, which is polymerase chain reaction.
And what is the polymerase chain reaction is?
Polymerase chain reaction was developed in 1993 by
Okay.
Kary Mullis,
Okay.
who won the Nobel Prize. PCR is a powerful molecular biology technique.
Okay.
Results in the rapid production
Okay.
of multiple copies of the DNA target sequence. The PCR technique has made it possible to analyze DNA fragments
Okay.
in samples that contain amounts of DNA that were either too small or too degraded to be
Okay.
analyzed otherwise.
Okay. And how does it work?
Well, what we did in the lab was we took 5 different things.
Okay.
The first thing that we used was a template DNA strand.
Okay.
The second was large quantities of the 4 nucleotides.
Okay.
The third was large quantities of the primer sequence.
Okay.
The fourth, DNA Taq polymers.
Okay.
And then lastly, the fifth was the necessary buffer for the reaction.
And what is the procedure? How they proceed it in the lab?
The PCR is divided into 3 separate parts.
Okay. What are those?
Denaturization,
I'm sorry. Okay.
denaturization,
Okay.
annealing, and
Okay.
extinction.
Okay.
The first is denaturization, which is conducted at 94 degrees Celsius for 1 minute. In denaturization, the DNA strand is heated
Okay.
until it separates or
Okay.
denatures. The second is the annealing of primers, in which a primer is used to anneal or stick to the DNA template strand where it finds a sequence complementary to its own sequence. This is conducted for 45 seconds at 72 degrees Celsius.
Okay.
Lastly, we did the DNA extension
Okay.
for 2 minutes at 72 degrees Celsius.
Okay.
In extension, the DNA Taq polymers is used incorporating, will extend the primer, incorporating the complementary nucleotide as it moves along the template strand, thus creating a new strand complementary to the template strand.
And why they use this technique for?
This technique is used for DNA fragments that were either too small or too degraded.
Okay.
So
It's amplified it?
yeah, and it amplifies it. For
Okay.
instance, we could find DNA just from a drop of blood at a crime scene.
Oh my God. I miss this lab.
Yeah,
What I'm going to do in order to get into this lab?
I would talk to the lab instructor. I don't
Oh
know.
my God. For heaven's sake. Okay, I'm going to talk to my instructor and I will ask him if he adjusts me into the next lab of this one.
Okay,
All right. Thank you very much, Alex.
I'll go with
I
you.
appreciate it. Yeah.
all right.
I appreciate that. You made my life. Oh my God.
And now 2 people are thoroughly happy that they enjoyed the benefits of PCR.